Now we will study the colloidal and the cellulose filters. These are very sensitive filters, and some colloidal filters can also be used to determine the viral size. The other filters until now we have studied, we can just isolate the filters and the pores, because of course depending upon the pore size, you can isolate the bacteria and you can also isolate the viruses. Most of them, the normal, you can retain the bacteria and the viruses can get through. However, the colloidal filters or the cellulose filters are very sensitive filters, and you can also use some other, just like the viral size, you can also coordinate with that. It is made up of nitrocellulose, and the pore diameter varies. Of course, it depends upon the experiment or the investigation which you are going to do, and you can determine the pore according to the experimentations or according to the virus which you are going to study. Variations is also there by the addition of some alcohol, ether, acetone or acetic acid which you can use, and you can also apply the electric field on these filtrations. You can just make it on the basis of the electrodes. You can isolate and you can differentiate between the viruses, and pore diameter can be 30 micrometer to 10 microns. And most useful in determining the viral size. Of course, we already studied that these are the very useful for the determining the viral size. You can see the in details here. These are two diagrams given here. One is just dialysis, the simple dialysis on the basis of which the molecules are differentiated from each other, and they are just differentiated on the basis of the just nitrocellulose which is present inside this. And then the electrodialysis on the other hand, here you can see here just the simple dialysis. We are using the nitrocellulose membrane here, which is the made up of the colloidal filter. This is its funnel, and this is the dialysis bag. Here are the solid particles which are going to use, and here are the ion which are going to crystallize inside the nitrocellulose membrane. Here are some other materials just like fresh water, or you are going to use liquids for this. And this is the just normal procedures of the dialysis for this kind of the filters. However, here it becomes the specialized and more sophisticated, and we are even able to see the viral size with such kind of the slides. Here are the ion which are going to crystallize, and we are already produced two kind of the electrodes on both sides, so that it become more sophisticated, and we can even use it for determining the size of some of the viruses. Then there is a cellulose filter, that was a nitrocellulose. Here we can use it the cellulose filtration method. It can be the pure cellulose or maybe any of the derivatives of the cellulose. It's coarse with the pore diameter, and the size depends upon the 3 to 7.5. While collodion filtration method, that size was from 3 to 10. However, it is medium with pore diameter and 0.75 to 7 micrometer also. It is dense with 0.2 and very dense below 0.2 micrometer. So of course there are different kind of the pores you can use. You can choose which kind of the pores which is important for your kind of the research and your kind of the investigation. Here the cellulose filter, which is just used for, and maybe you can use it in the cellulose, or maybe any other derivatives of the cellulose can be used for this filtration.